Okay, while it's coming on, I'll thank Pinaki and Arnab for inviting me here. And also, I will start with an apology. I realize that there are many people who have heard a version of this talk before, and even very recently, even just as of last week, but I hope to keep it even slightly interesting for them. So what I'll talk about is essentially the articulation of a physics experiment about a deeply important biological subject, and it relates to probing metabolism and energetics of living systems. All of this work is being led by a PhD student in my lab, Nishant Narayansamy. Some part of the work is being done in collaboration with a couple of biologists, Will Ratcliffe and Peter Juncker at Georgia Tech, and some other parts are being done with Pablo Sartori and his student Camila in Portugal. Okay, it's a bit of a truism. Metabolism keeps living matter out of equilibrium, of course, and broadly speaking, it's this flux of energy and material that gives rise not only to the form, but also maintains function as dynamic traits of non-equilibrium steady states. These examples are many. Some striking ones which relate to some of the community here relates to mechanics and spatial organization. I show you 3 examples over here. What you're seeing on the left is frog eggs which are taken and crushed, and the extract of those frog eggs is taken and put under a microscope, and you see this spontaneous self-organization, spatial organization of that cytoplasmic mixture. This is organization at the molecular scale. If, of course, these eggs would survive this crazy experimentalist, they would naturally undergo development. This is a frog embryo during the process of development. What this egg itself is about a millimeter in size, and what you're watching here is the spontaneous flows and rearrangement and reorganization of the embryonic tissues, which eventually give rise to shape. And once these frogs hatch, and there are many of these tadpoles, even at this population scale, you can get very interesting spatial organization, such as flocking of these tadpoles and so on. And indeed, over the years, it's thinking about phenomena like this at these various scales that has led to the articulation of this field of active matter. And it's not only sort of derived its inspiration and feeding back into explaining much of biological phenomena, but it's also given rise to a very rich program in synthetic active systems. One example that we just heard from Ambarish, and again, I won't go into the detail, but there have been various studies on these synthetic systems looking at these collective phenomena and so on, which are reminiscent of these biological dynamics or living dynamics. What I want to tell you today about relates also to aspect of metabolism driving the out of equilibrium nature of living systems, and it relates to growth and replication. As Jacob famously said, the dream of every cell is to become 2. What you're watching here is a movie of bacteria that are growing and dividing, right? So simply this is what you're seeing over here is that there is some nutrient in the ambient medium, and even if you put one cell over there, there's the flux of that nutrient which leads to the growth of the single cell, eventually to its division and giving rise to many more. So in my lab, what we have been working towards is sort of to try and couple the mechanics and spatial organization, which has been highlighted by this program of active matter for a couple of decades now, with the processes of assembly, replication, and ultimately even evolution. And we are, so I'm going to tell you a little bit of one sliver of this entire program that we are working towards. And what I imagine this to be is sort of a synthetic biology from a physics perspective, not from an engineering perspective, which people have been doing for a long time. And so let me dive right in into saying something about the energetics of metabolism. So the problem is as follows. There is a bunch of cells, unicellular organisms, if you want to think about it that way. There is some food, and during the process of this metabolism, there is increasing biomass, but it's also producing some waste. Okay? Ultimately, we want to ask if, of course, if we can capture the dissipation involved in such a process by something like this. And there's a huge question mark over here. I've just written something down, right? So what I've written down is the entropy production relating to the heat that's dissipated from this process, the chemical fluxes, and also something related to the growth itself. Okay? First, we want to even check if something like this is true for living systems. It seems like something like this should have been done by now, and the answer should have been known. Surprisingly, it's not. What people have been doing is sort of looking at various of these quantities in many different contexts. In some cases, people have been measuring the total heat release, in some cases fluxes, growth rates, and so on and so forth. And there is one data set which has come up from maybe a century of, or even longer, of such measurements, and has been summarized in this very curious, yeah. Sorry? So first of all, can we even, think of dissipation in these biological systems and measurably like this? And if that is the case, what differences exist in such dissipation across organisms, across functional states, across evolutionary trajectories? So many of these things have been speculated that the energetics or metabolism of living systems either constrains these aspects or enables these aspects, and so on. And we want to see in what quantitative manner does it do that.
How will you ever know whether where you get happy truncate that's feeding that?
Good point.
Some other machinery not going on.
Absolutely, you're absolutely right. Okay, so this is not something. There are some indications. For example, here you can have a look that this could indeed be true. Okay, we're working with that. You're absolutely right. Maybe that leads to more probing of the microscopic details and so on. All right. So many of these kinds of measurements have been made, and over the years have been summarized into a data set like this. This is referred to as Kleiber's law, if some of you recognize that term. What is being plotted on this is a log-log scale. On the y-axis is what is referred to as metabolic activity. And on the x-axis is the mass of the organism. And you see that this is a remarkably large range, going all the way from unicells to elephants and blue whales and so on. And there's clearly some structure here, and people have been endlessly debating what this exponent should be or what this exponent is. And in this particular paper, the case is being made for an exponent which is 3/4. Yeah, if it was something like 2/3, people make the argument of a surface area to volume ratio, but there seems to be a departure from that. And I'm not going to go into the details again, but there have been many theories and models trying to explain what this exponent is and why that is the case and so on. So one of them here, due to West, Enquist, and Brown, is that this 3/4 exponent can be explained because of an underlying fractal network which transports nutrients across the organism and so on. But clearly the data also goes down to unicells and things like that. Yeah, so the y-axis is quote-unquote metabolic activity. And thank you for asking this question. So there's no one single kind of measurement that has gone across all these organisms. In some cases, people have measured the heat that's emanating from an organism. In some cases, they measure oxygen respiration. Some cases, they measure the food uptake. Some cases, they measure biomass growth. And as you will see from this, all these have also been done at different temperatures. Clearly, you can imagine that what can be measured on a cat cannot be done on a mouse or an elephant. And when exactly should one make such a measurement? As soon as this organism is born, when it's an adult, as soon as it's fed, resting, so there are many complications. Yeah? And there are a variety of fudge factors converting one number to another, and eventually somehow all this data is put on this one plot. Yeah? So clearly there are many problems with this data set, and that deeply bothered me when I first started looking at this. But indeed, so let me just sort of summarize these caveats. The data has been collected from various sources, each in a different condition. There are a variety of fudge factors to convert data across measurement types. There is no precise definition for the y-axis. This last statement is a very interesting one. When I say selection pressures are completely unknown, people have speculated, even in building many of these theories, that either this energetics constrains evolutionary trajectories, or the evolutionary trajectory itself constrains metabolic activity, right? But how do you ever check something like that? Because we have one experiment that has run and random measurements that have been made.
So what is selection pressure?
Sorry?
Selection pressure?
By selection pressure, I am indicating the process of evolution, right? So we've had some in the deep past, an origin which has given rise to all this complexity, and all of this has emerged via some sort of selection or adaptation or and so on. That's what I mean by selection pressure. Okay? And again, there are a variety of theories, each explaining some or different features of the data, and it's very difficult to test these things in any systematic manner. In some, there is no experimental system. And what I want to tell you about is the articulation of an experiment for this thing. So what do we need from a toy experimental system? We want to have a range of organisms which span orders of magnitude on the x-axis. And because this argument about evolution has been invoked, we want to have known or controllable evolutionary histories for these organisms. And I'm making the job quite hard for myself. I want to have tractability to measure the y-axis in controlled standardized settings, various of these measurements. And I also want to have the ability to study the microscopics, like you asked. Are there many other factors that can come in over here? I want to be able to look at the genetic, molecular, biological, etc. So this is a very tall list of asks and claims, and I want to try and convince you that we are on to an interesting experimental system that might meet many of these needs. So I introduced to you this organism called the snowflake yeast. The snowflake yeast was evolved in the lab by Will Ratcliffe, one of my collaborators. It's a multicellular form of yeast, and as I will show you in a moment, it spans about 9 orders of magnitude on the x-axis in terms of range. What you're seeing here in this image, you see the scale bar over here. Each one of these is a single yeast cell, and these yeast cells are connected to one another via these attachments or bonds in the cell wall, and this is what makes it a multicellular structure. Okay, and I'll also try and convince you that this is an organism, not simply a cluster of cells. So how was it developed? So Will Ratcliffe, in this beautiful paper in 2012, so he was asking the question, multicellularity has evolved multiple times on Earth. So he reasoned that perhaps this is an easy enough transition to replicate in the lab. And so his motivation was to ask, can I go from uni to multicellularity in a laboratory experiment? So he started with single cells of yeast. These are about 5 micron or few microns in size. Typically in the lab, they're grown in a liquid medium under shaking. So this is a liquid culture that's grown under shaking. And what Will did was, after every round of growth to saturation, he would let this culture settle under gravity and then pick from the bottom and propagate those forward, but remain connected, all the cells remain connected eventually. But you might ask, what's the difference between this and a colony of yeast? Okay, a colony of yeast also does this, although in the colony of yeast, they don't remain stuck, but still it does produce a cluster. This cluster exhibits stereotype sizes depending on the selection pressure, right? So I can start from the ancestor and keep increasing. So what I've put on the y-axis is the generations of evolution. On the x-axis is the size. And you see that I've also put a characteristic or a stereotype size for this cluster. What do I mean by the stereotype size? Unlike a colony, these clusters divide. And this number that I've put here is the size at which the cluster divides. So the scale bar is the same on all these movies, and you will see that these clusters divide at a smaller size in comparison to this. And what is even more interesting is that if I take a single cell from this cluster, it will grow to a cluster of that size and then divide. If I take a single cell from here, it will grow to this size and then divide. So this macroscopic size is also encoded at the level of the single cell, and it's heritable. Okay? And now I want to call it a quote-unquote organism. I mean, this is, of course, Silka, yeah. Sorry, I couldn't hear you.
So do you know how this is selected? So what happened there mechanically? So why would it select a size?
I'll say a little bit.
Yeah.
I'll say a little bit in a moment. Yeah, surely. Yeah. Yeah, sure. So this macroscopic size, this multicellular cluster is maintained in the selection regime. If I remove that selection pressure, it's outcompeted by single cells, and eventually single cells fix in the population because there are growth rate differences. It's coming at a growth cost. Okay, so now, I mean, this is of course a huge debate now whether to call this an organism or not, but I will stick my neck out, and in my physicist way I will call this an organism for the following reason, is because the macroscopic object is the unit of Darwinian selection. What do I mean? If I take a cluster of a certain stereotype size, I can put it through two quantitatively different selection regimes. Yeah? In one selection regime, I give this culture 25 minutes to settle versus 5 minutes only to settle, right? So these are different regimes of selection, and this cluster responds by either dividing at a larger size or at a different smaller size. Okay? So it's in fact this macroscopic size which is under selection and not the single cell itself. And therefore, this cluster which is exhibiting already this property of division is also responding as the unit of Darwinian selection, and therefore I call it an organism. Okay? Yeah.
Why is that?
I'll tell you in a little more. Okay, so just to sort of summarize, this is a unicellular to multicellular transition that Will has observed in the lab. Increased size is a heritable trait. It divides as a multicellular aggregate, and also there's some determinate growth. In other words, there's some life cycle. What I just said, if I take a chunk of this cluster, it will grow to a certain size and then divide. It doesn't just break off at any size. Okay, so in the meantime, this experiment has been running both in Will's and my lab, and in the meantime we have clusters which are about half a centimeter in size, really big ones. And these are sort of microscopic fluorescence stained images of these things, and you will already see from these images that the morphology of the single cell itself has changed over the course of this evolution. It has gotten longer, right? And I won't go into too much detail of the mechanics, but I will very quickly say that it's not only the aspect ratio of these cells, but also the bond strength between these two that has been changing, which either increases its stability, which essentially increases its stability and therefore prevents fracture. And the fracture itself seems to happen because of building up of the stresses due to growth inside this cluster and also aging of the cells. And to your question of why it's growing in size in generations is because also the selection pressure is being continuously shifted to make them grow bigger and bigger. Okay?
So how homogeneous would be the size of your populations?
So these are the error bars that I showed you here. Yeah. So there's a distribution, but it's yeah. Okay, right. So what do we have? We have about 8 orders of magnitude of volume. I will tell you why volume and not mass in a moment. We have the full lineage and evolutionary history starting from the single cell all the way to the end, and I can rerun this tape as many times as I want in quantitatively different manners. I'll show you in a moment that we are setting ourselves up to measure the y-axis in careful laboratory settings, and yeast is a model organism for which all the genetics and molecular biology tools are available. All right, so what are we setting ourselves up to do to measure this y-axis? The heat production, we're using calorimetry to measure the heat production. We can look at the growth simply by very standard techniques of growth measurements in the lab. And these chemical fluxes, we are developing both microfluidics as well as mass spectrometry tools to measure these molecular fluxes. So we don't need to use picocalorimetry. These are large enough, and they're throwing out so much heat that any standard calorimeter can make this measurement. But we have a calorimeter that can give us sort of throughput. We can make many measurements at once. All right. But before going into these measurements itself, as you've been asking, increased size, of course, puts mechanical demands. One is about, this fracture aspect of it at all. And this is the argument of Galileo. Can a mouse be the size of an elephant? And there are structural constraints. I won't go into that. But another one that comes up is about transport or nutrient distribution. Yeast is an organism which doesn't have any flagella. It doesn't swim. And therefore, as it gets bigger, it quickly goes past this size where just diffusive currents are sufficient to feed or meet its metabolic needs. One way is that this huge mass of cells, it's just there to increase mass because it gives it a selection advantage, but really doesn't grow. That's one way of saying, I'm still diffusion limited, but I'm okay with that. But what we see is that these organisms, even up to this centimetric scale, grow exponentially when they are submerged in a liquid. But as soon as you take them away from this liquid environment and put them on a solid substrate like agar, this exponential growth goes away and the growth becomes linear, suggesting that a role for this ambient fluid medium. And what we discovered is that these clusters generate very strong and spontaneous fluid flows in the ambient medium. So this is the cluster, and what you're watching here are tracer particles that we have put into the ambient medium. And what you will also notice is that in one plane the traces are coming in, and in a plane out of focus the particles are going out. So this is sort of a circulatory flow that's being set up by these creatures. Okay. Just sort of looking at that same thing from the side, this is the cluster, and you'll see a plume or a fluid flow that's rising and circulating back. And I just want to make one quick comment here. We've looked careful EM and so on. There are no flagella that have developed in this evolutionary trajectory. Okay, but still these flows are actually, the flow speeds and so on are quite comparable to the flow speeds that are generated by multicellular organisms which have flagella, like the Volvox and Stentor and so on. And in fact, people have been making this argument for a long time that you can get macroscopic multicellular size only if you have flagella because of this aspect that you cannot beat diffusion limitation otherwise, right? So perhaps this is one example that we have uncovered here where that's not the case. Sorry, are there internal motions? There could very well be internal motions, but we think that there is a simpler way to get these flows. First, let me tell you that the flows are a consequence of metabolic activity. So if what I'm plotting over here is the mean square displacement for the tracer particles outside, and you get a flow only if the cluster is alive and metabolizing. If it's dead, nothing happens. Even if it is alive without any sugar metabolizing outside, nothing happens. Okay? And these flows are sensitive to gravity, suggesting a fluid density driven instability. Yeah? Indeed, yeast colonies have been shown to exhibit such an instability in the past. So let me first tell you what is the experiment. One of these clusters is stuck to the bottom over here, and we make a flow measurement in a particular plane with respect to this floor and the organism, and make the same measurement in the same plane by flipping the entire experiment, and you see that the direction of the flow is reversed, which means that with respect to gravity they're going the same way. It's just reoriented with respect, and suggesting that they are indeed gravity driven. And I can tell you a little bit more about the mechanism later, but there's also a size threshold beyond which only these flows emerge, below which there are no flows. Okay, so another very quick point. Because of the presence of these flows, these hydrodynamic interactions, as we've also heard in Ambarish's talk, give rise to interesting collective effects. So if I put many of these organisms, again these are of these various sizes, and if they're alive and generating these flows, you see that they spontaneously aggregate versus the dead ones, which really do nothing. And a very quick point, of course, much has been said about active phase separation, motility-induced phase separation being discussed in the absence of any long-range interactions and so on. But this flow-induced phase separation was something that we looked at with swimming droplets in the past. And there are other biological examples of this flow-induced phase separation, such as starfish embryo, which form these clusters of bacteria and so on. And we think that it's a very similar mechanism here. And I just want to make one very quick comment in the biological context, which is that it's a natural question that to ask, what is the biological function of these aggregates? And it's a meaningful question to ask in this particular case, because these are found in the natural world, and perhaps this behavior has been selected for and so on. Here is a unique example of a biological system which has never seen the effect of this collective aggregation in its evolutionary trajectory. All it's been selected for is weight. And so this presents us a unique opportunity, in my opinion, to try and ask this question of what kind of a cost function can I impose and under selection, and see what that does for the biological system itself. Okay. And if we look very closely, you see one of these guys zipping by. Indeed, flows plus an asymmetric object give rise to motility. So these creatures are indeed motile, and that's another story. They're motile and there's an interesting trade-off with growth and so on, which is also seen in other organisms. Coming back to these energetics measurements, yes?
No, it's really interesting. So I wanted to understand, because I mean the flow has to come from somewhere. So what generates this? It's not flagella. It's probably division. I assume is too slow. So where do they come from?
It's sugar uptake. So you're taking up sugar. If you're taking up sugar sufficiently fast, you deplete the region around you, and therefore you create a lighter fluid right around you. And then you set up a classic fluid instability, which
okay, so you say it's essentially just chemical gradients in
I
there?
think so. I think that's what's going on, yeah. As far as we have been able to measure, I think these chemical these gradient flows are dominating every other effect. Okay, so so now we want to embark on, and I'm going to just give you a teaser over here of what we're doing, and because I also made a comment earlier, in what condition do you exactly make this measurement? We want to carefully define such a condition. So we are starting with looking at making this measurement in a condition where these organisms are not growing, but still metabolizing, and also not producing this flow, just to keep things simple for a start. So how do we achieve this? First, we look for careful environmental, meaning food and other conditions, in which we can maintain these organisms and cells in a non-growing condition. So this is number of cells or organisms as a function of time, many days. And this number is really flat, in no death, no birth, they're just staying constant. And under such a condition, we can put them into a calorimeter, and you see that over many days, in fact, it just produces a constant flat heat signal. And as is to be expected from this, this is extensive. Thank you. So if you take half the number of cells or the quarter number of cells, it scales accordingly. And if you kill the cells, it produces no heat. Right? So from such a measurement, we are able to get the total heat that's being produced by these organisms. And as a very quick and dirty thing, even before we go into careful time-resolved mass spec and all the other things, we can simply measure the total amount of glucose when we start the experiment and when we end the experiment. And you see that indeed when they're alive and metabolizing, even though they're not dividing, they still spend some glucose. If we just compute the enthalpy corresponding to this change in glucose, it's reasonably close to what we are measuring from the direct heat release itself. Okay, I won't say much more about this. Still work in progress. But oops, what happened there? Okay. So just to sort of give you a quick feeling for what we are measuring over here. So what I'm plotting here, unlike in the previous one, this is volume, not mass. And this is also because the mass density is changing. It's right. So I think we should look at the scaling we should separate all these various factors out. The y-axis is this heat, and so I won't put a number on this, but I just have some guides to the eye for you to see that something interesting is indeed going on over here. We are now making measurements in conditions where we can turn back this flow on. We can get them into growing conditions, what have you. So stay tuned, and perhaps in the next talk I can give you some more news. Thank you.
So why did you make the choice of looking at volume, and what happens if you do look at the mass instead of the volume?
Right. In the previous plot it was mass because that's the easier thing to measure. You just put something on a weighing scale. You just don't have access to this information easily, right? And in fact, some of the theories suggest that the scaling could indeed come from storage. Right? And that's the reason why we also want to keep track of this change in mass density, right? And indeed, it changes quite significantly. Yeah.
Very nice. Just a curious point I wanted to make. Do you see in your cluster or sample some aging transition going on? Because they're active and the dead very quickly. Do you see?
You mean during our heat measurements? Yeah, that's a very good question. I don't yet know the answer to this, what is going on. But what we see is that even so typically we make this measurement over 5 days. And over these 5 days, this sort of remains just flatline, right? But we really haven't looked inside the cluster to see if all of them are living and so on. Sure, sure.
Yeah, just a follow-up on this question. So now when you're looking at the cells, like depends on at which growth stage the cell is in, right? If you're looking, like wouldn't the heat flow also change, and in which stage are you choosing?
So that's
it?
the reason why we started with non-growing. I mean, the cells are not replicating, okay? So we start with an initially exponentially growing condition, and then we sort of quench that state, right? And then we just keep it in that state for as long as this measurement can go on.
So in one of the slides you had mentioned that the growth rate is dependent on the ambient medium. So on solid it reduces, it becomes linear, and in liquid it becomes exponential. So I want to understand why it happens so.
Right, so in order to get exponential growth, it means that every part of this organism has access to nutrient and is able to respond in the same manner as every other. But if you don't have that, that's no longer the case. And therefore, if you're on a solid substrate which doesn't support this flow, then you deviate from that behavior. Great question. We don't know yet. I mean, it's very hard to look inside the cluster and make this measurement. What we know, of course, is that it has a certain branching architecture, which means we know how many buds one can give, and therefore from that get it. But during the later generations, what's happening is that they're getting elongated, and the number of buds is changing, and there's also entanglement and making it more complicated. Sorry?
Doesn't the mass density as a function of scale give you the answer?
No, it doesn't, because the single cell morphology is also changing, and the porosity is also changing.
The next speaker is Sunil.